I'd like to thank Natalie and James for organizing this awesome seminar series. I've been able to attend a bunch of them, and they've been awesome speakers, like Ishmael has been. So today, hopefully, I'll be able to talk to you about some of the work that was mostly done at Carnegie Mellon University, where I did my PhD, and then that we've since taken out to FluidForm as a startup company to hopefully develop it further. And so this talk will be focused specifically on bioprinting. So kind of thinking about how we can leverage bioprinting to design tissues for structure and function. And so I'll hopefully be able to talk to you about a different way of thinking about bioprinting, and then why I think that is an important way of thinking about it. And then how do we kind of grow as a field in terms of using 3D bioprinting for tissue engineering research, and then how do we move that towards clinically relevant research. So as a company, I think a lot of our thoughts on bioprinting was developed at CMU, where we think this can be boiled down to two key problems that we see must be solved for us to progress. First, I think if you're working in the field of tissue engineering or regenerative medicine, a lot of you are probably working with a unique material that has certain desired properties for a certain function. And this could be anywhere from an electroactive material that needs to be integrated with excitable tissues. It could be a tunable stiffness material that really allows you to control the microenvironment of the cells and tissues. However, most of these materials actually turn out to be difficult to print and assemble in 3 dimensions. And as a result, a second problem kind of emerges, where it's not really possible to fabricate a lot of these desired geometries that you might want to print to fabricate. And so kind of what we've done at FluidForm is now made it possible to now print these materials in any geometry. And here I like to show an example picture of a printed scaled down model of a human heart derived from an MRI file, where this is printed entirely out of collagen type 1. And I will share, hopefully be able to share a lot of this work with you coming up in this presentation. And so some of you might be asking, what exactly is 3D bioprinting and where did it come from? How come at every single conference now there's now a session on 3D bioprinting, and I'm seeing it at every single conference. So shown here are actually some of the more recognizable images, I think, of 3D printed constructs. On the left, you have this blue vase, which is printed out of a thermoplastic material. And so in a hot glue gun-like process, thermoplastic can be kind of melted and pushed out of a nozzle onto a platform. And then that molten thermoplastic is deposited layer by layer, and it quickly solidifies upon cooling to build an object. And on the right, you have these metal powders inside a container that can be melted by a very strong laser. And then all of this powder gets sintered together by that laser to then create complex 3D printed parts, as you see up there. And so all of these parts are produced using 3D printing processes that have been developed around the intrinsic properties of the materials. And for a long time now, a lot of people in the field of biomedical engineering have asked the question, could we borrow these same 3D printing technologies that allows us to print plastics and metals and use that to print biomaterials that are interesting to the human body? So the challenge is that these biomaterials are oftentimes very soft, and we're talking about a lot of these very familiar materials shown here on the left, including cells, collagens, fibrinogens, etc. And a benefit that plastic and metal printers take for granted is that the materials that they print are a lot of times far harder than the soft water-laden hydrogels that make up our body. So plastics and metals can be very quickly heated and cooled to form them into stable shapes. However, with protein hydrogels, that's not actually very easy to do. And so analogy that we make a lot is that is a watch assembling problem. So if I were to give you a disassembled Swiss watch, as you see here, you can probably rebuild this because each of the parts are, while they're small, they're all rigid parts. And so with some time, some knowledge, and some practice, you can most likely learn to put everything back together. But if you kind of imagine now that you have these protein hydrogels, and now that all these parts are soft, they become very hard to assemble, and you end up with this intertangled mess of clocks, these soft clocks that you see here in this really famous Salvador Dali painting, where everything drips all over the place. And so this is actually exactly what we see when we try to print a collagen 1 solution material directly into a petri dish onto a platform. This is supposed to be just a simple rectangular slab where the slab should have sharp edges, but everything here kind of coalesces and forms a big puddle. And I'd like to mention that each of the material to the left here really comes with their unique challenges when you think about handling them, for printing, in terms of rheological properties, their gelation mechanisms, and then also their gelation kinetics. So that's kind of the first core problem that makes bioprinting so difficult, where you have unique materials that are hard to print because they are so soft. And what we've kind of done as a field in terms of leveraging bioprinting for regenerative medicine and tissue engineering purposes is that we kind of have started to modify each material to make it more printable. So you can imagine you start with something that is hard to print, a soft biomaterial. You might think about modifying the material chemistry to tune its gelation mechanism and kinetics so that the moment it gets deposited onto a platform, it rapidly solidifies. So this can be achieved using a UV cross-linkable material where you shine a UV light right after you print the material onto a platform. You can modify the composition so that it has kind of the consistency, the rheological properties, so that it doesn't flow everywhere. It can stay in place, so you can stack everything together. You can also print additional scaffolding materials, kind of like how you would build a building, so that you use stiff structural elements to support your much softer material. And kind of each of these kind of limits some of the applications in terms of there's some drawbacks in terms of, for example, for the first one, you sometimes end up with reduced cellular binding domains and kind of increase or decrease hydrogel stiffness depending on how you're chemically modifying your material. When you're tuning the composition, you're a lot of times lowering the concentrations of the components. And sometimes you need to address the presence of these stiff structural elements as supports. You need to think about how do you remove them? Will they impact the tissue function? And because we kind of need to so finely tune each material so that they become printable, we usually don't have quite the perfect material. And when we run into this second problem where you have these desired geometries that actually cannot be printed with your materials because the materials are so soft. And we're in this in-between point where materials are somewhat printable, but they need to be printed in some of the simpler geometries designed around the challenges of printing them. So to summarize these two core problems that we see to bioprinting, one is you have these materials that are hard to print. Two, you have these desired geometries that are also difficult to print. And you might notice that we haven't really even talked about kind of the functional properties of these constructs. Usually, we like to, as engineers, we like to think about our design process as outlining the functional criteria first, and then picking the materials and geometries to create that thing. And unfortunately, I think right now we're kind of reversed in terms of how we're approaching some of this, where we're spending a lot of time making a material printable and then finding a geometry and then assessing the function of what's created. And so kind of very briefly, I just kind of want to have the audience go through a thought experiment with me. So if you were to imagine these problems that exist, what would you print? If you could now print anything with any material with the right geometries and then really start to explore the printed function or the fabricated function. So you might imagine, or I might imagine, you could start to replicate some of the full scale of the vasculature system within each organ. So starting from the smaller capillaries, building up to some of the larger arterioles, and then having these vasculatures then fill that volume of the organ. The bone is another example of different scales, architecture, and pattern that we really like, that really exemplifies the role of structure in function. And then also you could think about these composite tissues that involves different materials such as muscles, tendons, ligaments, and bones, and how all of that work together in concert. You have these valves that need to integrate with soft tissues around them. How do you address fabricating that valve so that it interfaces with the soft tissue and then does not elicit a severe immunoresponse? And then you could even start to dream up some of these more impossible, somewhat biomimetic looking, but nowhere to be found geometries seen here. So these are kind of the scientific questions that we are interested in studying. And this is why we kind of developed the technology platform that now allows us to print these soft materials in any geometry. And this is kind of the title of this talk, which is this fresh bioprinting platform, or short for freeform reversible embedding of suspended hydrogels. So seen here in embedded 3D printing, this is a type of 3D printing called embedded 3D printing. So we're not printing in air anymore, we're printing inside a support material. So seen here in this picture, you have this syringe loaded with a blue soft material bio ink or soft biomaterial inside a syringe. This is being pushed out of an extrusion needle, and this is your print. And then this yellowish material is the support material that is everywhere supporting your printed object. And what's unique about the support material is it is actually a Bingham plastic or a yield stress material. So this is very much has the same rheology or the same behavior as a, say, toothpaste. When you can squeeze it out of a tube, it'll flow out of the tube, but when you put it onto your toothbrush, it won't actually fall off when you turn it around upside down. So this yield stress behavior comes from the fact that the support material is derived from these millions of gelatin microparticles that get packed in very tightly in our process, and then they act to provide the support material with this yield stress behavior. So I think this process is really simple and intuitive when you watch it happen. So here we are printing a blue alginate material, which if you're not familiar, it will cross-link very rapidly in the presence of calcium ions. So we have calcium ions inside the support material in yellow, and when we start printing, you can see that the alginate ink is being deposited layer by layer. We're printing the letters FluidForm. And then while it's printing, the calcium ions in the support bath begins to cross-link the hydrogel so that it solidifies, and then you can then raise the temperature on this entire system to melt away the gelatin microparticles. And this is really what allows us to print for function with any jelly material and any geometry. And then so this technology was initially described in 2015 in Science Advances. All of this was done at CMU. We followed this up in 2019, published in Science. And then this was then what led us to spin out the company where we've now at FluidForm. And so in these next slides here, I kind of want to show you some examples of how we utilize this technology fresh for creating some of the structures that we wanted to create. The first example I want to show you is this idea of orthogonal gelation mechanisms, or just printing of different materials that gel in the presence of different, or they have different gelation mechanisms. So for example, collagen will gel when neutralized from an acidic condition to a neutral condition. Methacrylate HA is the methacrylation, makes it light sensitive, so it will cross-link in the presence of UV light. Fibrinogen is enzymatic. It will be cleaved by thrombin to turn into fibrin, and alginate, we've described previously, is ionic. And so what's unique about the support materials, you have these gelatin microparticles. They're surrounded by this interchangeable fluid phase. So what is really interesting is you can actually substitute this fluid phase with anything you want as long as it's compatible. So you can include a pH neutral buffer, you can have the UV lights on top, you can have thrombin inside the solution, and you can also have calcium ions inside the solution. And so when you put all four together, you can actually achieve a 4-material print simultaneously. And then so here you have the colors matching those colors of the material in the letter CMU here. And in terms of some of the basic building block features that we can print using Fresh, we took collagen type 1 as an example material to explore this, because as we all know, collagen is the most abundant protein in the human body, and it really has this really large footprint in biomedical research for us to understand its role in biology and disease. So we started with looking at individual filaments, where we were able to recreate 200 micron filaments down to 20 micron filaments. We can laterally and vertically stack them, pack them, and also very easily vary the spacing on them, and then begin to utilize all of these to then assemble these filaments into macro scale structures. And so taking these printing capabilities, we want to see how well all of this holds up inside a large print over multiple length scales. So we chose a heart as a proof of concept model because that was what we were interested in, was cardiac tissue engineering. And so we derived an SCL model from an MRI file. We printed this out of collagen type 1 directly into the support bath using FRESH. So this is seen here as a printed heart model. If you take a look at the cross-section of the print, you can see some of the finer detail structures, such as the trabeculae, that's really important in development and contraction, and then also some of the infill structures that we've incorporated to fill in the walls of the model. So in this micro CT image, you should be able to appreciate some of the finer details. So you have the ventricles shown here, the trabeculae, the rectangular infill, and then we were also able to print the valves, the pulmonary and the aortic, and then you have this chamber walls that really surround and provide the heart with this structural integrity. And the heart, of course, was just a scaffold. It didn't have any particular function other than just a demonstration of the printing capabilities using FRESH. So we kind of set out to really think about how to recreate function in some of the key components of the heart, with the first one being the valve. So we printed the valve again. You can see in this video that we're able to handle the valve, and the leaflets remain patent and they are open. And then in this next video, we put this valve into a very simple perfusion flow loop setup and show that the valves open and close as kind of they're intended. But of course, this is very early on preliminary work where this is in no way optimized for function. And so the kind of next critical piece, the puzzle of engineering heart tissue, is that the heart, like any other tissue in your body, really requires this dense network of vasculature that supplies blood to its cells. And so one of the challenges that we run into is that when we take MRI data, a lot of times it doesn't have quite the resolution we need to give us those finer details, such as capillaries. So what we decided to do was to computationally generate these vasculature networks. So what we did here is that we segmented out a section of the heart, which is in here, the left ventricle. We took the left ventricle, utilized the larger branches to then use as seeding points to generate some of the finer, smaller vasculature networks. And then so we then isolated this smaller portion of this lattice for printing, shown here, and this is the printed object. We then took this print and optically cleared it so you can better see the perfusion of this print. You can see that here we're flowing just a very simple glycerol water solution through each of the channels, or from the larger branch down to the smaller, which range from 2 millimeters or several millimeters down to a couple hundred microns. And so this is really exciting for generating and creating some of the smaller, or the multi-scale vasculature system that you find. And then the last piece I want to show you is, of course, the key functional component to the heart that allows it to contract, which is orchestrated by cardiomyocytes. So here we create a very simple model of the left ventricle. So here, this is a very commonly used model for depicting the ventricle, where within the walls of this model, we printed 2 collagen shells that kind of surround the central region, that's printed using a very highly concentrated solution of cardiomyocytes derived from embryonic stem cells. So this was printed using a 300 million cells per ml solution to get that really nice dense network of cardiomyocytes. And we're interested in doing this because we really wanted to see the cardiomyocytes electrically and mechanically couple so that they can start to contract more effectively, so that hopefully we'll see some meaningful, appreciable contraction in this 3-dimensional structure. And what we also saw was that the cardiomyocytes really begin to align along the direction, circumferentially along the direction of the ventricle wall. So very quickly, I'll show some of these calcium imaging data that we found, which really highlighted the presence and the uniformity of this calcium propagation really highlights the uniformity of our printing. You can see the same thing happening above. We can also take a point stimulation probe and measure out some of the conduction velocity metrics that we're interested in. And so this model really is, again, exciting to us because it's really the first step to developing more complex three-dimensional models that we can very finely tune and control in 3D space. And so I'll kind of show you this, this next slide here very quickly to highlight the work that we're now doing at FluidForm. Is we've taken this capability and printed some of the other structures that are more commonly found in injury. So we're very commonly used in medical devices. So heart valves there's been a large need for a long time, which we've shown before. The ACL is, of course, a very common injury site for athletes and just for everyday folks like us. And then the trachea, as well as the intervertebral disc for spinal injuries. And so in all of these examples, you can see that these can be handled. They kind of maintain their desired functions, and the regions on these prints can be controlled for stiffness, for their structure, so that they can eventually lend to function. So we've been very excited to see that a lot of people have adopted Fresh as a platform and started to publish with it, with growth year by year. And then we've really taken on at FluidForm this mission of expanding the reach of Fresh, making this as easy to adopt as possible, starting with the powder, getting into a dish, and being able to utilize this platform for any material on any geometry, any printer, and any software. And the idea is that we can't think of any or all the applications out there, and it really needs to rely on the community to develop some of the interesting questions that we want to study. And so I'll leave my email address here. These are some of our collaborators, and feel free to reach out if you have any more questions. And I want to emphasize that a lot of this work was done at CMU with awesome collaborators that really contributed to a lot of the work shown here.
Thank you very much, Andrew. That's a great talk. Whilst we wait for some of the questions to come in on the chat group, I was going to start with a basic one just for my understanding. I wondered, how are you getting from the print that's within the gelatin to then the structures you showed just on like a couple of slides before, whether then the kind of the fully formed like regions?
Yeah, so for example, these prints were printed out of collagen type 1. And so collagen is typically an acidic solution that will gel when neutralized, and also when it reaches or further cross-links or further gels when it reaches body temperature. So what happens is that you would take this acidic solution, print it into the support material. Inside the support material, you have a buffer that very quickly neutralizes the collagen solution. And then so as you're printing, you're now beginning to solidify these gel or collagen filaments that we then stack layer by layer to create the three-dimensional structure. And then at the very end, you just raise the temperature on a platform or put it on a hot plate that's around 37 degrees Celsius to melt away that gelatin. And then you're left with a cross-linked or gelled structure that you've designed and created and printed.
Excellent, thank you. I think James had a question as well, so I'm just going to quickly pass over to him.
Sure.
Yeah, I was just wondering about the support material and whether there did need to be a degree of tuning for different materials or different sort of concentrations of materials. So would you be able to use the same support material for a very, very soft collagen solution and a very, very stiff collagen solution?
Yeah, definitely. So we spent a lot of time kind of like playing around and developing the support material while when we're developing this. And it certainly requires a certain rheology to be able to embed and trap your material. But within that range of rheology, you can print different collagen solutions of different concentrations. The challenge is that after you release it, is the material property of that low concentration collagen that becomes somewhat problematic at times, because now you have this really soft hydrogel that can very easily, it's hard to handle. You have this very wet tissue paper like it's really soft collagen material. Even though it can be printed in a three-dimensional structure, it will very easily fall apart when you try to, say, seed cells on it, or you just kind of push it around with the spatula. So I guess the answer is that it can be printed, and then it's just kind of developing strategies to then think about how you would process the final print.
Perfect, thank you.
Thanks, Andrew. So we have a question from Ishmael as well, who said, great talk. Could you comment on the remaining channels on the ACL ligament, for example, to be ready for testing in animal models? I think he's like asking what steps are now required.
Yeah, I think the most obvious next step is testing of the mechanical properties. And of course, the ACL is a very complex structure. It is not just a simple, soft, stiff material, right? And it is not just made purely of collagen type 1, even though the majority of it is. But all of the different composition of materials is what lends its unique properties of being able to twist and bend and then be able to extend and withstand your everyday sports or actions. And so the next steps will be to start to tune some of the material compositions, start to think about different ways of printing the structure, so you can align some of the print filaments so that they in the direction of force loading, and then start to test the mechanical properties, say starting with the Enstron with very simple stress strain curves. And so, but it's definitely interesting to us. And there's also a lot of post modifications to the prints that can be made in terms of cross-linking, where you can add chemicals such as glutaraldehyde to treat these. And so those are some of the next steps that we're taking and interested in taking.
Excellent, thank you, Andrew. So we have time for one more question from Akash. So thanks for an interesting talk. For printing orthogonally gelling materials simultaneously, can different inks be injected from separate nozzles? Also, if multiple gelators are added within a single ink, can uncross-linked components be removed following gelation?
Yeah, I think the separate nozzle question is it's definitely a yes. It depends on your setup, your system. It gets somewhat tricky to arrange different nozzles to print different regions of the print. But that is something that is also fun about it, is how do you design the print so that it's now is more complex. I think the second part of the question was, can you remove the uncross-linked material? Is that correct? And the answer would be yes. If it's uncross-linked, it will just kind of diffuse out of your print. When you kind of release the whole thing, you can wash it away, and it really is, again, up to you to design the experiment. So a lot of this, I think, is really very similar to how you would approach tissue engineering, fabricating different 3D gels for cells. It's really, we've kind of added the component of a 3D printer to now arrange all of this in 3 dimensions.